Hey, this is Sam Hutton. I'm the tomato breeder here at University of Florida, located at the Gulf Coast Research and Education Center. And with me today is Dr. John Smita. John and I wanted to present some research on breeding for bacterial wilt resistance. So pieces of the puzzle in pursuit of bacterial wilt resistance in tomato. John, you joined my group here in 2018, so can you tell us a little bit about where you're from and about what you did prior to coming to Florida?
Yeah, hey Sam. So before I joined Dr. Hutton's group, I was up at Cornell working on my PhD there with Dr. Martha Muchler and working with tomatoes and some disease resistance breeding. And before that, I was working at the University of Missouri where I did my undergraduate in plant sciences. And before that, just growing up on a vegetable garden. So I've been around vegetables a lot and plant disease and have always been interested, and it just seemed like a good fit to come work with Sam. And so thanks for having me here, Sam.
Yeah, well, it was a pleasure. You jumped right in and you hit the ground running. It was great because I just handed off this project to you. This is an NSF funded project, and our overall goal with this project was to identify broad spectrum disease resistance for bacterial diseases in tomato, and to leverage this in crop breeding. Right. So here in Florida, bacterial wilt can be a significant disease, especially up in the northern part of the state. Not quite as much down in the peninsula of Florida, but it's more of a problem North Florida and up the East Coast into Georgia and the Carolinas, Virginia and Tennessee. But here, it's bacterial spot is a very, very significant disease for us. So even though this project is focusing on bacterial wilt resistance, we hope to apply some of these results to improve our breeding efforts for bacterial spot disease. I think it's interesting to point out that in my own breeding program, we have some really high levels of resistance to bacterial spot. And coincidentally, all of these sources of resistance have some common ancestors in their pedigree to our bacterial wilt resistance sources. So some of the material that John's going to talk about today is actually in the pedigree of a lot of our bacterial spot resistant breeding lines. I don't think that's a coincidence at all, and that's why we're working on this. Now, with regard to bacterial wilt, this disease is a really big challenge. It's a worldwide disease problem, and the pathogen, Ralstonia solanacearum, is very complex. Lots of different strains, and those strains behave differently and sometimes react differently to resistant sources. Right? Resistance in the plant is controlled by multiple genes, which really complicates our breeding efforts. And resistance is also associated with reduced fruit size, and this further complicates our breeding efforts, especially here in Florida, where we're breeding for large fruited fresh market tomato. Right? The point of all that is to emphasize that we really need a better understanding of the genetics of bacterial wilt resistance in order to make further progress in our cultivar development efforts. And this is where you stepped in, John. You conducted some really large experiments here in order to start answering some of these questions. So can you tell us a little bit about the trials that you set up?
Yeah, so one of the difficulties with bacterial wilt is the need to do very large screens to accurately phenotype for the disease. And so everybody in Sam's program is very optimistic that we don't need to do any more screens this size. Almost 20,000 plants in this trial. And as you can imagine, that's a lot of effort. And we did this experiment 3 different seasons, 4 times total, twice in one season, to get consistent disease pressure and enough replication to tell differences between the plants. When the plants are small, it can take about a day to rate the entire field, but when the plants are larger, like when we took this video, it can take several days to rate the plants for disease. And during the course of an experiment, we rate the plants weekly or biweekly until the susceptible control has most plants that are wilted or dead, which usually is about 3 or 4 times in a season. And here's a video kind of showing a plot of healthy plants during the course of a disease trial. And you can see there's a lot of dead plants, missing plants. We had plots of 4 plants here. And so this disease, bacterial wilt, causes wilting. The telltale signs of wilting, as you can see from this plant here, which is caused from the disease getting into the xylem of the plant and loss of turgor pressure. And so that's the telltale wilting that we phenotype during the course of a disease trial. Here's an example of what some plots look like toward the end of a trial, you can see some plants here that clearly died early on, and so you can see the devastating effect of having this disease take out plants early is you basically lose all the yield. The next plot here is a plot of healthy plants, so a more resistant recombinant inbred line here, and it has larger fruit, which is closer to the size that we're trying to get to while maintaining the resistance. And so the bacterial wilt resistant donor source, this Hawaii 7997 control, which is shown right here, has very small fruit, but high levels of resistance. And so that's one of the large problems we had to overcome, is trying to get to fruit that are a little larger, like this, while still maintaining high levels of resistance. So we're not quite there yet. We're trying to get to fruit that are a little bit closer to this size and quality, and we're getting close to that point, but we're not quite there yet.
All right, so John, you mentioned that in these trials you tested 2 different populations. So what did you find out about the resistance in either of those?
Good question. So these populations, the BW2 and BW3 population, they both derive their resistance from that Hawaii 7997 control, which we showed you in the previous video. And you can see from the 2 graphs above that Hawaii 7997 control is on the far left side of the graph, showing that very few of the plants of that control actually wilted during the course of what's depicted here, which is a trial we did in the fall of 2019 here at the station. And the BW3 population, which is shown on the bottom, was originally derived from Hawaii, but the direct parent of the recombinant inbred line is Florida 8109, which has larger fruit. But unfortunately, you can see from the distribution of the recombinant inbred lines that most of them are skewed to the right, which means that there was a lot more wilted plants, and so less resistance in that BW3 population. Whereas the upper chart, the BW2 population, which was derived from Hawaii, the direct parent was Florida 6, which had higher levels of resistance. You see the distribution is more uniform, and that there were a lot more recombinant inbred lines that had higher levels of resistance. And so we're going to focus on some other trials we did and some QTL mapping results with that BW2 population for the rest of this presentation. And we did test a total of 161 recombinant inbred lines from the BW2 population in those 4 different field trials, as we mentioned. And we also genotyped each of those lines with a panel of molecular markers across all 12 of the tomato chromosomes. And those results were used to help determine which regions of those chromosomes contained bacterial wilt resistance genes. And so this chart here that we're looking at is a QTL mapping result, which is shown from one season in fall 2020, showing the significance of certain areas of the tomato genome that are associated with resistance to bacterial wilt. So the height of the peak there represents how significant or how involved that peak is, that region is for resistance. And the colors there, we actually looked at 3 different time points during the course of that experiment. But you can see that chromosome 6 peak is very important for resistance to bacterial wilt. And so are these kind of in line with what we were expecting to see, Sam?
Yeah, I'd say so. First of all, we knew that we had multiple genes involved with resistance, and that's what your results are showing, and that's great. And then we also knew that some researchers in Asia had previously identified a couple of QTLs on chromosomes 6 and 12, and your results are matching right up with those results. So we didn't know that chromosomes 6 and 12 would be important for resistance to the US strains of bacterial wilt, but we did expect we could find them, and now we're finding them. So now we see they're important. We see that chromosome 6 is particularly important, so I'll certainly be selecting that in my breeding efforts going forward. The chromosomes 2 and 3 loci we didn't know about, and so this is great. This is really useful information for me to help guide my breeding program. Right, so 4 QTLs all total. What you didn't show here is that your other population, that BW3 population, identified an additional QTL that we did not expect before. And so now that we say there's 4 to 5 QTLs that I need to be selecting for. John, were you able to figure out if any of these QTLs may be associated with fruit size, and if that might explain one of the challenges that breeders have had over the years?
Yeah, so great question. That's definitely one of the challenges with breeding for bacterial wilt, is often there's an association with small fruit, like from that resistant source, the Hawaii. And so we screened the BW2 recombinant inbred line population using a replicated number of plants and looking at a bunch of fruit for their size. And we saw, as you can see in this QTL chart here, that there are 3 regions associated with smaller fruit that we detected. 2 of them on chromosomes 4 and chromosome 7 are not associated with any regions that are important for bacterial wilt resistance. So that's good, and hopefully those regions are easier to eliminate and increase fruit size. The 3rd region on chromosome 12 that's associated with smaller fruit is on the same chromosome as some resistance QTL that we mentioned. So that might be a little bit harder to eliminate the small fruit QTL and maintain that resistance QTL on chromosome 12. We also did some follow-up validation experiments with some of the recombinant inbred lines that were segregating for one of these regions, and it helped to validate that these regions are involved in smaller fruit in this population. So where's this project headed next, Sam?
Well, John, you did a tremendous amount of work on it. I really appreciate that. You generated a lot of useful information that I can use in my own breeding program, and other breeders can utilize as well. When you start thinking about future directions for the program, I think it's clear that I need to address that chromosome 12 QTL that's affecting fruit size, right? So if I'm selecting for bacterial wilt resistance, that QTL is going to be coming right along with my resistance unless I can break that linkage there. So that'll be something I can do, and we can fix that. It'll just take a very focused effort. Another thing I want to do is look at those 5 QTLs that you identified. You've got some information that speaks to the magnitude of the effect of each one, but I need to confirm which ones are really essential for achieving bacterial wilt resistance, a field level of resistance. If I don't need all 5, then I'd rather not select for all 5 QTLs. All right, so which ones are the most important? Which ones give the greatest effect for field resistance? I need to do some work to figure that out. All right, along the same lines, I need to understand how each of those genes work. If some of them are dominant, then I may only need to incorporate that resistance QTL into one of my parents for a hybrid. But if the gene action is additive, then I may need both parents to carry that QTL. So obviously that definitely directs my breeding program and how I'm going to select for these QTLs. Not to mention, lastly, that I would like to know which of these have an effect and what is the effect against other bacterial pathogens, in my case, bacterial spot called Xanthomonas. So we'll wrap it up with that. Just by way of acknowledgments, thank you, John, for all the work that you did on the project. I know it was a lot. There's a picture of our lab. They were really key in getting these trials accomplished. They loved getting out in mid and late July and planting a 20,000 plant trial. They're sad to see you go, John. You just left my program. You're no longer a postdoc with me. Where are you going now?
Yeah, so I'm continuing to work with vegetables, working for a vegetable seed breeding company called Earthwork Seeds, and continuing to work some with tomatoes and peppers and other things, and just happy to take the things I've learned here and take that to my new position. So just thank you for the opportunity and everything that I've learned here. It's been a fun project, and I'm sure the lab will be sad to see these trials come to an end.
Yeah, well, thanks for your work here. Hopefully we have opportunities to collaborate more in the future. Other collaborators on this project, Gary Valade has helped with the trials that we're conducting here. Dr. Matthews Parrott in North Florida and his student, his prior PhD student, Dr. Sanju Kunwar, did some work on these populations prior to John joining my lab. And Jay Scott helped me develop these populations early on, and he actually worked with United Genetics to get these trials evaluated in India. We didn't share the results of those, but those are really interesting as well. Stark Air Seed Company, located in South Africa, did some testing with these materials there with their local strains. And I have to acknowledge again the funding from NSF to get this work done. So thank you all for joining in. Hopefully we got some time for questions afterwards.